separated. So the neurotransmitters that we have talked about, one is acetylcholine, it's very common. It is secreted between the muscle and the neuron, okay, where the motor neuron connects to the muscle. That's where we get acetylcholine, and it has to be broken down after, for such that you have relaxation. And in this case, it brings us to the fact that they may not ask you about synaptic transmission. Instead, the question can be explain neuromuscular transmission of impulses. The concept is basically the same. Okay, the concept is basically the same, and we shall look at that shortly. But the neurotransmitter in that case is acetylcholine, and after using it, it is broken down such that it does not cause more excitement. And the more excitement that you have for the muscle, the more it contracts, and you don't want to have that. And then we have some amino acids, glutamate, glycerin, and GABA, as other transmitters that are secreted. Yeah, like we said, that some synapses can be excitatory, others can be inhibitory, and the explanation remains the same. And then we have some synapses that do not have channels. We call them non-channel synapse. That means that the transmission is not through the neurotransmitters that we have talked about, but through chemical messengers. And in this we talk about adrenaline, dopamine, serotonin as some of the examples. So this is what I talked about, the neuromuscular junction. The mechanism of transmission across the neuromuscular junction is basically the same. Arrival of animals in the presynaptic node causes calcium channels to open. This results into the fusion of the vessels, synaptic vessels, with the presynaptic membrane, and then releasing the neurotransmitter that diffuses across the synaptic cleft and attaches to the membrane called the sarcolemma. And this membrane is for the muscle, and this causes excitement that will result into an action potential. The action potentials in the muscle will cause the muscles to contract. Electrical synapses, that means that you have the membranes connecting, membranes of the two neighboring cells connecting. That means that the impulses move from one cell to the other without the need of neurotransmitters. And this is what you can have in the heart and the eyes. This means that one of the disadvantages of synaptic transmission is that it delays. It is not as fast as with the electrical impulses. So there is a delay in chemical transmission across the synapse because you have the neurotransmitter having to diffuse. So what are the properties? We have one summation, and that summation can be spatial or temporal summation. What does spatial summation mean? Simultaneous release from multiple presynaptic neurons causes cumulative effect on the postsynaptic neuron. The meaning of this is that you have several presynaptic neurons that are connecting to a single postsynaptic neuron. So you have very many of them, and they're connecting to one which is post. That means that if you simulate, if you have simulation of these individual presynaptic neurons, that is going to be, the stimulation is going to be summed up such that it is very easy to excite the postsynaptic neuron. That is summation. Summation is about adding up. Okay? And then temporal. When we talk about temporal, the word temporal is associated with time. Time is associated with temporal. So temporal summation. If you stimulate a presynaptic neuron repeatedly in a short time, with a short time interval, it means that you're going to increase the amount of neurotransmitters that is being released, and therefore you will have action potentials being released. This is basically to say that if you stimulated not frequently, you have a time lag in your stimulation, it could not be easy for you to have an action potential. But if you stimulate repeatedly in a short time frame, then it is very easy for you to have an action potential. So that's temporal summation, that the individual stimulations that you are making would not result into an action potential, but because they are repeated in a short span of time, they are going to add up such that the threshold value is reached and you have an action potential. Repeated stimulation of the presynaptic neuron will result into production of the neurotransmitter, yes, but the amount will actually decrease, and that is what we call adaptation and fatigue. Okay, it basically means that the presynaptic neuron gets tired of releasing the neurotransmitter. That is synaptic fatigue and adaptation. Facilitation. If you have an impulse that has passed, having a second impulse to pass, if it is an exercitory postsynaptic potential synapse, then it is going to be very easy for you to have the subsequent impulses being able to pass. That is facilitation. That means that the passage of the first impulse increases the sensitivity of the postsynaptic neuron, making it very easy for you to have another action potential in the postsynaptic neuron, even when you have weaker stimuli. Inhibitory signals make it very difficult for you to excite the postsynaptic neuron, even if you have reached the threshold value. So you will not fire an action potential. Okay, so generally, synapses are important for transmission of information. In this case, it is chemical transmission through a neurotransmitter. The other thing is that the transmission across the synapse is in one direction, unidirectionality. You cannot have the impulse across the synapse moving bidirectionally. One of the reasons is because you have the acetylcholine being broken after it has been used. That means you don't have acetylcholine again to excite this other presynaptic membrane. Okay, but also the lack of these neurotransmitter receptors or the presynaptic membrane means that you can't have backward excitement. And then the other thing is that, yes, you have the impulse that is arriving at the presynaptic. That point is about amplification. Yes, when the electrical impulse arrives at the presynaptic node, it is in the electrical form. And then you have it being transmitted across the synaptic cleft through chemical transmission through the neurotransmitter. But the point is that the excitement in the postsynaptic neuron is actually going to be higher. That is what we call amplification. Why? Because of the properties that we have talked about. The summation which is temporal, And then the summation, which is also spatial summation. And these are what is responsible for the amplification. Integration. And you will have many synapses, of course, even in the brain. And this allows coordinated responses to be made. So synapses are important in integration and information. Let me organize this. The spacing, look, the last slide. Okay, so synapses can enable what we call integration, where you are having information coming from different points, from different inputs. And if information is coming from different inputs, yet you need a coordinated response, the synapses actually help in that. The other thing is that synapses can filter weaker signals. The meaning of this is that if the threshold values are not reached, then you will not have excitement. So therefore, you will not have the synapse responding to any stimuli that is extremely weak. It only focuses on strong stimuli. So that marks the end of this presentation about synapses. Thank you.